Okay, hello. This is the KD and thermodynamics of protein ligand binding and a little bit on cooperativity video. So at the end of this video, you should be able to define KD, so it's a vocabulary word, determine the KD from a graph, interpret or analyze KD, and verify in thermodynamic terms. This is going to involve math. Woohoo, finally here. If you don't have a calculator with you, you might want to get a calculator out just so that you can practice as I'm going along. Define the three types of cooperativity. Describe a myoglobin oxygen binding curve. Describe a hemoglobin oxygen binding curve. And just in general, interpret protein ligand binding curves. Ligand is also a vocab word in this video. Some people pronounce this word ligand, so you may have heard it before. I pronounce it ligand. Some have pronounced it ligand. Food for thought. Okay, so this is part 3 of our 4-part story, cooperativity, thermodynamics, and KD, which all has to do with how oxygen is reversibly transported throughout the bloodstream without being chemically altered. And we'll only have one video for this part 3, and then there'll be one last part 4 video, and then we'll be all set with the oxygen transport. Okay, so I wanted to do a really quick recap from last time. We had to start by finding a way for oxygen to solubly travel through the bloodstream and be reversibly bound without being chemically altered. So the first thing that we talked about was adding iron, which binds oxygen super well, too well, way too well to be reversible. We bound it to a porphyrin and covered up 4 of its 6 coordinate covalent sites. And then we covered the 5th coordinate covalent site with the proximal histidine, which left 1 site open for oxygen to bind, O2 to bind. Now that histidine had to be part of a protein. The protein is globular to increase the solubility of oxygen transport, and it turns out a single subunit, which would be myoglobin, is still not great at reversible binding. So our last thing did is we added some more subunits to introduce allosterics and increase reversibility. So all this going from left to right is all about increasing reversibility of O2 binding and increasing the solubility. Let's just put solubility there. And then we also talked a little bit about the equilibrium between the T state and the R state and how that is dependent on 2 things. One is enthalpic stabilization, with the T state being stabilized by salt bridges, and the T state in this diagram here are the boxes, and then the R state being stabilized by the coordinate covalent bonds and the hydrogen bonds when the oxygen is bound, and that is the R state, the circles. So enthalpic stabilization. The other thing that affects the other piece that affects the concentration, so how much of the T state versus the R state, is the concentration of oxygen. So as you increase oxygen concentration, again, Le Chatelier's, we push more towards the products on this case, and that's the R state. If you decrease oxygen concentration, we come back to the left. Oxygen should be released or unbound, and we get to the T state. Okay, that was all review from before, so now we're going to get into the quantitative description of protein ligand binding. Ooh, it sounds super fancy. It's super cool. It's a very important concept and quantity that biochemists use. So in biochemistry, or I should say biochemists, we don't just like to ask the question, does it bind? So does oxygen bind to myoglobin or hemoglobin? Because we really typically only get yes or no answers. What we really want to know is how well does the ligand bind? Okay, and with this question, you need to answer it using numbers, or you need a quantitative description. So if we can put values, numbers on binding, we can compare the binding if we have something, some sort of control. So before I move on, let's define the word ligand or ligand. This is a molecule. It can be large or small. Sometimes ligands are even proteins. You can have a protein-protein interaction. One of them would be called the ligand. In our case, when we're talking about oxygen transport, we're talking about O2 being the ligand. It's a molecule that binds, what do I mean by binds? Maybe associates with another molecule. In our case, that's a globin, or the protein. Okay, so that's the protein ligand. Associates with another molecule and is not chemically altered. So a ligand is not changed into anything else after binding. So a substrate in a reaction might be changed into a product. In this case, the ligand comes in as O2, and when it unbinds, it is still O2. It has not been chemically manipulated. Okay, and the number that we're going to use, the value that we're going to use for our quantitative description is going to be called K. That's a capital K and a lowercase d. This is also called the dissociation constant. Okay, so consider the following. You have a protein, and I'm going to do Pac-Man protein. You have a protein, and it binds to, or you think it binds to a ligand. That's going to be my ligand. And there's some sort of equilibrium between the unbound, where the two parts are separate, and the bound. All this protein here, and the ligand is now attached through non-covalent interactions, maybe even a covalent interaction. So that is bound. We would also call this PL, representing the protein ligand complex, and over here this is P plus L, because they are unbound. They are not together. The kinetics, or the right kinetics, that's a lowercase k. I tried to write them a little funky so that you can tell the difference between my lowercase k and my uppercase k, has an on rate and off rate. Okay? And KD mathematically can be defined by either the on and off rate or the concentrations of products and reactants. So I'm going to give you the two equations, and we're not going to derive them, but you do need to know both of these equations. So KD mathematically is reactants over products, because it's the inverse of the association, or you can represent it in terms of it's the off rate over the on rate. So you just need to know these two equations. You will likely not be given them on an exam. The definition, man, spelling is rough, of KD. So this is the definition. This is what it is. KD equals the concentration of ligand, that's concentration of ligand, when 50% of the protein is bound. That is its official definition. So if you were asked for the definition of KD, this is what you would write. This is the definition. We also called KD the binding affinity. That's something else that we might call it. Okay, a quantitative measure. Now 50% is just a number with which we can compare all the other proteins to. So we need to be able to have something to, sort of a reference point with which to compare all KDs. And so you look at 50% binding and reference the KD back to that. And I'm going to show you next how you determine the KD. Okay, here we go. How to find KD from a graph, which is what you'll be asked to do on the exam. I typically don't give you values for the concentration of protein, concentration of ligand, concentration of protein-ligand complex. Instead, I give you graphs, and you have to analyze the graph. So here's an example graph. It goes from 0 to 1 on the y-axis, and this is sometimes referred to as little r. What it really is it's percent protein bound, oh, that's handwriting, with ligand, where 1 represents 100% of the protein is bound with ligand, and 0 represents 0. And then we have concentration of ligand on the x-axis. And this graph that we're going to look at is going to be hyperbolic, where it plateaus off at 100%. You can't have more than 100% binding, so that can't go above 1. And then down here, there will be a variety of values. I'm just going to make some of them up. 5, 10, 15, 20, 25, 30. You get the idea. And then concentration units on ligand, I'm going to say, are micromolar. You will need to be able to move between micro, nano, we actually don't go beyond nano, but you're in micro, nano, milli, and so on and so forth. You need to be able to move through those very quickly in this class, or be able to convert between micro to nano, micro to milli, and so on and so forth. So if you haven't, you might want to spend a little bit of time reviewing that. All right. Here's how we do it. Okay, so KD is the ligand concentration when 50% of the protein is bound. So first, we're going to find 50% bound. So that would be on this graph, 0.5. Figure out where, draw a line over until you hit the curve, and then drop that down to the x-axis. Okay, so where this comes down and hits the x-axis, that is your KD. So roughly here, my KD is 5 micromolar, because remember, KD equals the ligand concentration. Okay, so 5 micromolar represents ligand concentration because it's on the x-axis when 50% of the protein is bound. Okay, so I'm going to do that one more time. So if you're looking for KD, you're going to find 50% protein bound first. Figure out where that is on the y-axis, drag it over until you hit the line. And then you're going to drop that down until you hit the x-axis, and that is where your KD is. Now we're going to talk a little bit about what does this even mean, and what do I do with this number once I found it. So this is what I call the meaning of KD. Let's imagine that you have a particular protein. Again, I'm going to do Pac-Man here until we get to other proteins. I'm going to call it HK. And you believe that HK binds to glucose. And I'm just going to represent glucose with a G. It just binds to glucose. So there is an equilibrium between the unbound and the bound. I'm going to put G right there in the binding site. The area where the ligand binds on the protein is called the binding site. Just something to know. Okay, so let's say that you believe that HK binds to glucose. All right, so this would be a hyperbolic graph. 0 to 1. I didn't. Oh, come on. Didn't quite get all the way up to 1, did I? All the way up to 1 here. Okay, where glucose would be on this axis because it is the ligand, it is the thing binding to the protein. And we've got a series of values here. I'm going to put this in nanomolar. This 1, 2, 3, 4, 5, 6, 7, and so on. And again, this is percent bound. If I go 50% is about here, drag it over, and drag it on down. It's hard to see. That makes this 0.5 nanomolar. Okay, so what does that mean? So all of this is measured outside of the cell. KDs are measured outside of the cell where you have just the protein and you have just the ligand and you combine them, and there's a number of different techniques you can use to find the KD. So how is this even relevant? So let's say that you're given, or you know that inside the cell, the concentration of glucose is 2 nanomolar. So the question how about wood? Would HK and glucose likely be bound inside the cell? Okay, so here's how you figure that out. All right, so I know that inside the cell, the glucose concentration is 2 nanomolar. I have determined experimentally that my KD is 0.5 nanomolar. So at 2 nanomolar, it looks like I've got about, almost, I'll say 100%, oh no, 100% of HK, zoom out, bound to glucose. So yes, this would likely be bound. These two would likely be bound in the cell. By cell, I mean the thing with the lipid bilayer and the nucleus on the inside and the ER and things like that. If it turned out that, let's say, the glucose concentration was 0.1 nanomolar, 0.1 nanomolar is over here, and it looks like that would be, man, this is hard to tell on my hand-drawn graph. I don't know. Let's say 15% binding, 15%. This would mean about approximately 15% of HK is bound to glucose, and this would likely not occur. Statistically unlikely that these two things would be bound inside of the cell. The other thing that you can do, even let's say we have 2 proteins. You have the protein called HK, and you believe it binds to glucose. And it's reversible. This indicates reversibility. But you think that there might be also another protein that it binds to, and I'm going to call this one PK. You could find the KD for both of them and figure out which one binds better. You would need 2 graphs. 1, 0. Put the little R there for simplicity. Glucose is still your ligand, so it goes on the x-axis. I'm going to give them both nanomolar. If you'll notice, a KD always has the units of molarity because it is the ligand concentration. So please, 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 please do not forget your units. Don't forget units. Don't forget your units. I tend to take off a fairly good chunk of points if you forget your units. They're super important. Okay, so 1, 2, 3, 4, 5, 6, 7. we'll do 10, 20, 30, 50, and so on. Okay, so here, 50%, bring it over, drop it down. Okay, because of the way I drew my graph, it looks like the KD on this graph is about 1 nanomolar. And 50%, bring it over, drop it down. Let's say that's KD is about 18 nanomolar. Now, which one binds glucose better? Smaller KD equals stronger binding affinity. And there are actually a number of protein ligand interactions that have been quantified. HK. And there are some guidelines about what constitutes strong binding versus weak binding. So if the KD is less than 10 nanomolar, that's a relatively strong interaction between that protein and that ligand. If the KD is greater than 10 nanomolar, then it's considered weak binding. So if you look over here, you can see that the weakest binding is between the calmodulin and the calcium 2+. And then up here, the biotin and the avidin, those are the strongest binding. So high affinity are really low KD values, and weak is high. So it looks like enzyme-substrate interactions tend to be a little bit more weaker. Antibody-antigen interactions are about in the middle, but closer to the higher affinity, and so on and so forth. What does this mean, though? Like, how can we translate? Is there any way to translate this into thermodynamics? All right, so can we relate this KD in terms that might make it easier for you to understand that a lower KD is stronger binding and a larger KD is weaker binding? So let's do it in terms of thermodynamics. Okay, since we're going to mostly just be considering enthalpy. We're going to mostly just consider enthalpy. There is an entropic seesaw between getting the ligand bound and releasing all the waters, and yet constraining the movement of the ligand by bounding. So let's just consider enthalpy here. When the protein and the ligand interact, the interactions between the two are mostly always non-covalent. So you might consider that between the ligand and the protein, you'd have H bonds, salt bridges, ion dipoles, pi stacking, etc. So every interaction between the ligand and the protein is going to lower the enthalpy. Enthalpy, that's the delta H one. If the enthalpy keeps coming down, and let's just say entropy is a wash, it's not exactly a wash, but let's say it's about a wash, like it's about a tie, then if H is going down, and we're going to say that we're not going to consider entropy in this case, but if H goes down, then G goes down. So this indicates a strong interaction or strong binding between protein and ligand. Mathematically, we can prove this to ourselves. So KD is the inverse of KA. Remember, these Ks are capital Ks. And there's this nifty equation, delta G equals negative RT ln Ka. When we consider KD an inverse here, this gets translated, this gets manipulated to delta G is RT, so we lost the negative side, ln KD. So let's do the math for the two that I gave you before, the two examples that I gave you before. So where we have the HK protein plus glucose is in an equilibrium in a HK with G, and the other one is PK plus glucose in equilibrium with the bound form. And this one we found the KD to be about 18 nanomolar from the graph, and this one we found the KD to be approximately 1 nanomolar, and I said that this one is stronger, or it's winning, right? It should have, between glucose and hexokinase, or HK, hexokinase is HK, there should be lots of non-covalent interactions. But you can prove this to yourself by doing the math. So delta G equals, R here is 8.314 joules mole Kelvin. Temperature, I will usually give you temperature either in Celsius and you'll convert to Kelvin, or I'll give it to you in Kelvin, but we'll do 298 K natural log 1 times 10 to the minus 9 molar. So that will be for HK. And then for PK, I'll just do it underneath. We'll keep it in blue though, so hopefully you can see that. 8.314 joules mole Kelvin. Temperature is the same, 298 Kelvin. And here we have natural log of 18 times 10 to the minus 9 molar. So this is the point where you could stop the video and get your calculator out and make sure that your math checks out with my math. One thing to note is that these units cancel out and these units cancel out, so the ending units for your delta G should be joules per mole, joules per mole. You can convert to kilojoules per mole, that's fine, but it still should be joules per mole. So that should be your unit. So I changed my color. Can't have that. Joules per mole. Joules per mole. And I'm going really slow in this video because I want you to just make these connections and be able to do this math. All right, so the number that I got here is negative 51. That's a 5. Yikes. 51.3 kilojoules per mole. And the number that I got down here is negative 45.6 kilojoules per mole. So hopefully this helps prove to you that a lower KD is a stronger binding, which would translate into a more negative Gibbs free energy, which would also indicate that there's lots and lots and lots more non-covalent interactions happening between HK and G than there are between PK and G. Yikes. Okay. I hope that that made sense to you. You'll have some practice with this on your assignments. Okay, so now we got to go back to hemoglobin and myoglobin. So in this picture right here, the dotted line is myoglobin and the solid line is hemoglobin. And what you'll see on the y-axis for this is something called YO2. Some students start calling this YO2. YO2, it really is the same thing. It just means percent bound. And again, it goes from 0 to 1, 1 being 100% bound. Now on the x-axis, this is our concentration of ligand. Our ligand is O2, and because it's a gas, the units are torrs, okay, which is a pressure unit because it's a gas. So it's not in nanomolar, micromolar, millimolar, it's in gas units. Food for thought. Okay, so when I look at this, I can see that myoglobin has a KD. I go to 5, and I didn't drag that down quite straight at all. Myoglobin has a KD of, I don't even know, that's really close to, let's say 1 torr. And hemoglobin here, it doesn't quite work out the same when the graph is not hyperbolic, so you can see that there's an S shape here. That makes this graph sigmoidal. That's the S-shaped curve, whereas myoglobin's shape is hyperbolic. Okay, so if we say this is approximate of a KD, I'd say that's, I don't know, 28 torr, right? So what this is telling you is that myoglobin binds oxygen way, way, way better than hemoglobin. The other thing that this tells you is there's this tissue thing and the lung thing. So the tissues in your body, this is the range of concentration for oxygen in your tissues. This is the range of concentration for oxygen in your lungs. So what I see when I look at this is that myoglobin comes along and at super low oxygen concentration sponges up all the oxygens, and all the myoglobins end up being bound to O2. So at low concentrations. So at O2 concentrations that are lower than in tissues even, all the myoglobin proteins are bound. With O2. The sigmoidal shape curve indicates cooperativity. Okay? Cooperativity is when the binding sites talk to each other. So myoglobin has 4 binding sites. Hemoglobin has 4 binding sites. Hemoglobin has 4 binding sites. So there's 1 subunit, there's 2 subunits, there's 3, there's 4, and each one of these there's the heme, right? And oxygen, O2 can bind here, 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 and here. So it turns out that hemoglobin has positive cooperativity. Which is when, cooperativity is when the binding sites I'm going to say talk to each other. Positive cooperativity means that after the first ligand binds, the affinity affinity increases for the other sites. Okay, so how is this read on the curve here? So down here for hemoglobin, there's low affinity and none of the 4 sites are bound. Now as we increase oxygen concentration, the first oxygen will bind, which rapidly increases the affinity for the other 2, and then the 4th oxygen will bind, and then the hemoglobins will be completely bound with oxygen. I'm going to say that one more time. At low oxygen concentration, there's almost no oxygen bound to the hemoglobin. After the first one oxygen binds, the second 2 bind very rapidly, and then the 4th one binds, and then you've completely saturated your hemoglobins. Okay, so the sigmoidal shape curve in this case indicates positive. And the reason that we have positive cooperativity is because of allosterics. The change in shape is the communication between the 4 subunit binding sites. So when the first oxygen binds, the iron moves into the ring. The iron is connected to the proximal histidine, which is connected to a helix, which is connected to the protein, which is connected to all the other subunits, and the shape changes. And that shape change allows the next 2 oxygens to bind quickly, and then that 4th oxygen comes in last. So that's what positive cooperativity is. Myoglobin, the hyperbolic shape curve, is no cooperativity. So we can have positive cooperativity, we can have no cooperativity. That's where the binding sites don't talk to each other. And then negative cooperativity is the binding of the first ligand decreases the binding affinity for the other ligands. So in the last slide, we had positive cooperativity, negative cooperativity, and no cooperativity. Again, this is the binding sites talking to each other. In order to have cooperativity at all, you have to have a protein that has multiple binding sites. So you must have So on this one we have, this is one site, this is a second site. If the binding of the first ligand increases the affinity, makes it easier for the second ligand to bind, that's positive cooperativity. If the binding of the first ligand decreases the affinity for the second one, that's negative, and you can have it where it doesn't matter, they bind independent of one another. So this one may bind first, and this one may bind second, but there's no increasing or decreasing a binding affinity for the second, third, however many other ones. The reason I've drawn it like this is to show you that a single subunit can have cooperativity as long as it has multiple binding sites. Myoglobin only has one binding site. There's my heme. My oxygen can go here. Myoglobin only has one binding site, so it can't have cooperativity at all, and therefore its oxygen binding curve is hyperbolic. And this is a figure that I found that goes with this YouTube video. If you want to, you can take the chance to look at it. But down here is where the muscles are. So hemoglobin will be mostly unbound, which is what you want. You want it to release oxygen in the muscles, and you want it to gain the oxygens in the lungs. This is the R state. This is the T state. Again, those are dependent on oxygen concentration. So as we increase oxygen concentration, we should move to the R state. As we decrease oxygen concentration, we should move to the T state. Why does that happen? Enthalpy and Le Chatelier's principle. So at low oxygen concentrations, none of the subunits are bound with oxygen. Once you bind the first one, it increases the affinity for the second and the third, the second and the third ones to bind. There's an equilibrium here. The second and the third ones to bind, and the fourth one actually is the hardest to bind, has to do a little bit with entropy. There's less sites available. But this is just an interesting diagram of showing the sigmoidal or the positive cooperativity. And again, that positive cooperativity is facilitated by the allosterics. Remember, allosterics is other shape, so a change in shape can either kind of open the protein binding site up so the oxygen gets in easier, or closes it so it can't get in. So the allosterics can affect cooperativity. Okay, so a couple of food for thoughts. Do all proteins have quaternary structure? Nope. Your example is myoglobin. Do all proteins have tertiary structure? Not necessarily. but a protein has to have either tertiary or quaternary structure. So a protein that doesn't actually have tertiary structure is collagen. But it's still a protein because it has quaternary structure. Hopefully that's not confusing. Can a monomeric protein display cooperative binding? Yes. Monomeric means it only has one subunit. As long as it has, yes, with the caveat. As long as there are multiple binding sites. Okay. Do all proteins with quaternary structure display cooperativity? Nope. Not requirement. Are all proteins globular? Nope. For example, collagen is not. Do all proteins display allostery? That means they all have changes in shapes? Nope. Does a cooperative protein also have to be allosteric? So do you have to have allosterics in order to be cooperative, or cooperative have to be allosterics? This is not necessarily, but is typically the case where the cooperativity is facilitated, yikes, by allosterics. Okay, and that's the end of this video. So we've gone over protein ligand binding in the quantitative description, which is KD. That is the value that we compare all protein ligand interactions against each other. The smaller the KD, the stronger the binding. You can prove that to yourself, which the lower the KD, the stronger the binding, the lower the Gibbs free energy, which all indicates the interaction between protein and ligand is having a lot of non-covalent interactions. Interpreting the myoglobin hyperbolic binding curve tells you that myoglobin is really good at sopping up or soaking up oxygen, but it is terrible at releasing it, whereas hemoglobin is really good at binding oxygen at low concentration, or binding oxygen at high concentrations and releasing it at low. And I think if I look at my notes here, that's all I got. I will see you in class. Bye-bye.